very rudimentary schematic of what could happen after spinal cord injury. So you have on the left here is represented the brain, and the cell A is projecting from the brain to the spinal cord. In our case, if we're studying the cortex, that would make it a corticospinal neuron. That's a system that we like to study in the field of spinal cord injury. It's required for voluntary movement in you and I, and for skilled movement in animals. Again, focusing on the movement because that's largely what the field of spinal cord injury has. Now, if there's a spinal cord injury, there's a disruption, there's an interruption of this circuit. You get this secondary degeneration. These little purple macrophages come in. They're not actually purple. Come in, and you end up with degradation and retraction of axons, and then a formation of a scar around the injury site. So there's not any significant spontaneous regeneration that occurs after spinal cord injury. Right? There may be some level of local plasticity, and what we think is that that local plasticity really can underlie some of the gains that can be made through intensive rehabilitation and the progress that can be made, up to a year or two after injury. So there's that level of spinal shock in the first few months that can resolve some level of dysfunction. But we think that with really intensive rehab and weight-supported training for stepping, things like that, that individuals can recover, maybe taking advantage of spared circuits such as this sort of bypass pathway, areas of the spinal cord that are intact around the injury site. I mentioned that 50% of these cervical injuries are people who have incomplete injuries in some level of function. That may be motor function. It also could be that there's this plasticity effect and a growth and a rewiring of cells that are injured. Okay. So, great. So that's sort of the basis for what we're thinking of in terms of spinal cord injury. So what are the scientific breakthroughs in spinal cord injury research? And that's the part that some of you may be interested to hear about. First, I need to take a step back. Again, Dr. Anderson Erisman, now as president of the North American Spinal Cord Injury Consortium, prior to an NIH meeting called SCI 2020 in early 2019, sent out a survey of questions to individuals living with spinal cord injury and their caretakers. This is a newer survey, and asked them, to agree or disagree with certain statements related to spinal cord injury research. And one of them that I think is very important is that people living with spinal cord injury are tired of media hype about research results that are not yet available treatments. And perhaps unsurprisingly, a large number of individuals with spinal cord injury strongly agree or overall agree with this statement. There's very few that disagree. So a lot of people, they don't necessarily want to hear about how fantastic it is that a researcher can get a rat to walk again. They want to know about treatments that are going to be available in the clinic, something that's going to make a difference in their lives. And I think, it comes down to a few different reasons. Avenues such as this one are critical for engaging the public, communicating our research, and putting it in perspective of what our research findings mean and how they might shape clinical treatments. And, as a researcher, we're very privileged to get to do something that we love to do and ask scientific questions. But in order to do so, we have to convince funding agencies that what we're doing is important. And part of that is proving that what we're doing is innovative and novel and really hasn't been done before. And that is problematic when you're trying to get at something that you have to reproduce previous results and really have to show that something is safe and effective for recovery. People want novelty, and that hindered the field of spinal cord injury for a long time. A lot of people looked to what was the next molecule, what was the next IN1 antibody, what was the next thing that was going to drive this massive regeneration, and how they thought about recovery. And so, it's sort of an uphill battle in that when moving treatments from the lab, from the bench to the bedside, it goes through a series of steps of clinical research that are based first and foremost on safety. So some of you may be familiar now through the development of vaccines for COVID. You start off with smaller groups that are really looking at the safety first. And then you go to a smaller study that looks at both safety and some level of effectiveness in a small cohort of individuals. And then you move on to these large stage 3 studies in huge populations to look at overall effectiveness and things of that nature. And with something like a spinal cord injury, you have a lot of variability in the injuries themselves. You have a diverse population and things might not always translate. So there is a difference between a significant research finding and changing clinical practice. And I think that's something that we have to keep in mind. As primary researchers, basic researchers, we love what we do. Otherwise, we wouldn't do it, right? It's not something that is particularly profitable, especially in a wealthy area such as Westchester. So it's something that we really love to do, and we think that what we do is the coolest stuff on earth, and so we need to tell other people about that. But we also have to understand that people don't want to hear about how exciting it is that a mouse or a rat can wiggle its foot. Again, they want something that's going to move into the clinic, and so I'm gonna touch on why this sort of hyping things like this could actually have a negative effect. And I'm going to pick on stem cells a little bit. We heard about stem cells from Dr. Sani in one of these webinars a few months ago. And I don't mean to be overly negative, but I just want to run through, why this is something that we have to really keep in mind when we communicate our science. So stem cells are incredible. They hold great promise in that they can become several different types of tissues. You can make a neuron out of a stem cell. You can make bone. You can make hair. You can make skin. You don't necessarily want to make all of those things if you put them into somebody's spinal cord. So you want to be able to restrict their fate and get them to incorporate into the injured spinal cord to provide some level of function. And that's really not a trivial proposition. In California, when I was out there for a number of years in graduate school, there was a program, a state bond that paid for stem cell research and overpromised in terms of what would be delivered in 5 or 10 years from that. It was going to cure juvenile onset diabetes. It was going to cure spinal cord injury. And cure is a really tricky word there. So it allowed people to make great advances in stem cell biology, but to promise that it was going to have such a robust effect is really disingenuous. And it's something that is important to do. So with stem cells, people have put them into the spinal cord. Here's a study from 2005 about stem cells from the olfactory mucosa. These are autografts. These are from the same individual then that are getting the stem cells. And in summary, they say that this procedure is safe. This transplantation is minimally detrimental. It's really the general risks of surgery, infection, things like that they have to worry about. At the end, though, they say long-term patient monitoring is necessary to rule out any delayed side effects and assess any further improvements. It's really not a ringing endorsement, but okay, so we wait and see. All right, this is 2005, so 14 years later, we have one of these individuals, intramedullary cervical spinal mass, so that doesn't sound so good, after stem cell transplantation using an olfactory mucosal cell autograft. Mass lesion, so that's an injury to the spinal cord here. Again, here's another MRI, and we can see where these arrows are. There's a large mass that developed in this individual. There's a 38-year-old male that was referred to the neurosurgical clinic with a 3 to 4 year history of deteriorating neurological function in his arms and a gradual loss of bladder function. So this is somebody that was hoping to regain more function after injury and ended up losing more because of this growth of the mass, and originally had a C5, C6 spinal cord injury at 20 years of age. And so there's risks that, hyping these things and then potentially people seek out treatments that might not be well controlled. So stem cell tourism is actually a thing that could be very detrimental there was a study this year published on complications from stem cell tourism in neurology. So individuals that would report to their primary care physician, people that maybe told them that there wasn't much hope for recovery, and they wanted to seek out alternatives, somebody who would be willing to tell them things they wanted to hear. However, these complications can be quite severe. So tumors, infections, stroke, death, continued degeneration like we saw, sepsis, seizures, hepatitis, meningitis, blossom eating, I don't know what that is, myelopathies, encephalomyelitis. So these aren't all for spinal cord injury. These aren't all complications from spinal cord injury studies, but really moving things from the bench to the bedside is a process that is focused on safety first and foremost and needs to be very well controlled. And that's why these studies in the US are sort of few and far between. So that was a little bit of a downer. I'm going to bring you back. We're going to go now to the scientific breakthroughs in spinal cord injury research. I can't see the chat or anything, so I'm not sure if there's questions, if anybody really wants to chime in. Oh, and some of those spinal cord injury studies, these are things that people have to pay for up front. So this is, tens of thousands of dollars out of their pocket without any real evidence for improvement, $25,000, $50,000 at a time. So, even if it doesn't do something bad, it might not do anything good. So what are the breakthroughs in spinal cord injury research? I'm going to focus on just a small, I can't cover everything. I'm going to focus on just a small bit of research that's being done in individuals with spinal cord injury at the moment. So electrical excitation of spared spinal circuits. So by giving some current to the spinal cord, the nervous system works on electrical currents, and the activity within neurons is conveyed by ionic potential differences, so basically electrical currents within neurons, and then chemical connections or synapses between cells. And so I'm going to talk mostly about the work seated by Reggie Edgerton at UCLA, who has looked at this for 40 years at the ability to activate this circuitry in the spinal cord. The spinal cord is not simply a number of wires, of axons of passage, right? It's a complex computer. And what he's found in his research is that by activating this computer, by providing some level of baseline excitation, then these spared circuits can actually engage some of the spinal cord. And so this is really characterized by the work now of Susie Harkema and Gregoire Courtine, two different labs. These are people that trained with Reggie. What I'm going to show you first is a video where they've implanted a spinal stimulator on the cord and provided pattern stimulation that's meant to mimic the type of activity you would get with walking. So to pattern it in order to activate different populations of cells that drive walking. Here's an individual. He's got a harness on. He's got 65% weight support. There's a schematic on the left of the stimulator over the spinal cord, and then on the right, that schematic of the spinal cord is going to light up when it shows where the activity is. Now, you can see when they turn on the stimulator and these pattern stimulations, he's able to do weight-supported walking, and then there's activity of different muscle groups as it's being stimulated. There's a few examples here from this study. So you can see the individual is taking steps. Here's one where they're going to turn off the stimulator after a few steps, and you'll see that he can right here, and you'll see that he can no longer initiate voluntary stepping movements. So they turn the stimulator back on, and now he can start to engage those muscles again and step. And people, with this stimulator can modulate the behavior. Here's another one where they turn it off again, and this individual cannot walk. And then here's one where they ask him to make some exaggerated steps, and you can see that he will lift his leg up high as he steps. So these are voluntary movements in some, individuals that have no movements for a long time. And, with hundreds of hours of therapy and these stimulations, some of these individuals could do assisted stepping at the end without the excitation. All right, so I already told you that walking maybe isn't the priority. So now I'm going to move into the cervical spinal cord, and these are individuals between Chet Moritz and Reggie Edgerton that have cervical spinal cord injury, and now they put a stimulator on the surface of the skin on the back of the neck. So there's not a surgery involved, not implantation, and you're going to see somebody practice their eating using utensil. So at first it starts without any stimulation. Let's see, did my video go? Yeah. And you can see in this first session, the individual has quite a bit of difficulty leaning forward, trying to get the food to his mouth, practice food. Here's this first week with the stimulator on, the movement's a little bit more smooth. He can lift his hand further to his mouth. And then now we're looking at session 20 after 4 weeks. And this movement is much more smooth and much more capable. All right. So, what do these therapies do? Are they simply activating spared pathways and allowing people to re-engage them? Or are they actually changing the injured spinal cord? Is it possible that they're driving new connections in the spinal cord? And so for that, we have to look to animal models. And, my lab studies mice. We work in rodents. I've worked in rodents for my whole career. This is a privilege for us to be able to work with animal models. This is not something that we take lightly. And mice are not the same as humans. Some people move from animal models to non-human primates. We're not at that stage at this point. There's a pathway illustrated here. This is the corticospinal pathway that we study. You can see that it's different in the rodents than it is in non-human primates. It still projects from one side of the brain to the other side of the body. So left side of the brain controls the right side of the body and vice versa. But in primates, then this pathway has moved, I don't know if you can see my cursor or not, hopefully, from the dorsal columns here, from the dorsal part of the spinal cord through evolution, it's shifted to the lateral. So there's some anatomical differences. In primates, these cells make direct connections onto motor neurons, and that drives dexterous movement. So our colleague, Yutaka Yoshida at BNI, has made a mouse that retains these direct connections, and these mice actually have greater dexterity in their digits with this direct connection, which is interesting. So our former colleague, Jason Carmel, along with Jack Martin, has shown that through exciting this pathway, they can actually reorganize the projections of the corticospinal axons in the spinal cord. So in order to do that, they used an animal model where they removed 1/2 of the corticospinal pathway to the spinal cord. And now with the stimulation of the cortex, that's able to engage the mechanisms of axon growth and regeneration and elicit, and elicit more growth here in the spinal cord across the midline. So some levels of plasticity that can occur. Our colleague at BNI, Jian Zhang, has demonstrated that without direct electrical current, using a magnet and to drive current in the brain can do much the same thing, and is hoping to translate this into human studies. So really impressive work. We've been focused a little bit on the mechanisms, the molecular mechanisms that underlie this electrical stimulation. So I'm going to talk about work from my postdoc, Sebastian my mouth's dry. I told him I'd try and pronounce his last name properly. Hara, there we go, close enough. And we're interested in the peripheral nervous system, and I'll tell you why in just a moment, because we are talking about spinal cord injury. But the peripheral nervous system can regenerate after an injury. So after damage of the periphery, you'll get an induction of regeneration-associated genes in the neurons that project through the peripheral axons. So in the sensory neurons that are within the what's called the dorsal root ganglia here in blue, and in the motor neurons in red within the spinal cord, you'll get an induction of a number of genes that have been coined regeneration associated since they correlate with this regeneration. This is one called activating transcription factor 3, which isn't that important right now, but what you can see is in a whole dorsal root ganglia that's been cleared, there's nothing in the intact animals, but after 2 days after nerve crush, there are a large number of these little red dots. These are neurons that are expressing this transcription factor, and then you'll end up with regeneration. This regeneration is relatively slow. It takes about 1 millimeter a day, which you can imagine in a mouse is maybe not all that bad. In a week or two, you'll have regeneration because things don't have to grow that far. But in a human, it's a very real problem. Something from the shoulder to the hand can take 12 to 18 months, and so what others and what we've shown as well is that electrical stimulation of the nerve intact will actually drive some of these regeneration associated genes. Will activate this regeneration pathway that people like Bernice Grafstein and who's here with us today are really instrumental in demonstrating that there is this induction of a regeneration program. And so with electrical stimulation now, you'll get an induction of these genes and you'll get more rapid regeneration. So why am I talking about peripheral regeneration when we're supposed to be focused on spinal cord injury? Well, we have a project. So now I'm being a little bit selfish and I'm talking about our work. We have a clinical project where we're working with a neurosurgeon at Massachusetts General Hospital named Justin Brown, and he is trained in neurosurgery and plastic surgery, and he does peripheral nerve repairs. And one of the surgeries that he does is something called a nerve transfer. In that case, you can see in this beautiful illustration by Sidney Agger that after a spinal cord injury, there's a region of the spinal cord that is no longer receiving input from the brain and brainstem. And so these peripheral axons that were once blue are now gray, meaning that there's connections there. There's motor neurons and sensory neurons that connect to the periphery, but there's not information being conveyed from the brain or brainstem. If you take one of these or some of these axons that are functionally redundant, so you have many muscles, for instance, that mediate flexion of your elbow. You have your biceps, your brachialis, your brachioradialis. What he does is he takes some of these redundant nerves and he cuts them and he grafts them to nerves that are affected by the injury, those that arise at or below the level of injury and have intact motor projections. And then what that will allow for is some level of hand and arm function to occur. Okay, so this is a bypass. So here I've added to my figure. Now we have the periphery here, this neuromuscular junction. That's where the axons innervate the muscle and release acetylcholine. And so after the spinal cord injury, again, we have our interruption of the circuit. And now if we rewire, if you look on the right side here where it says PNS for peripheral nervous system, if we rewire that periphery and transfer these functionally redundant donor nerves, we can get some level of reinnervation of the recipient nerve targets. So now we can push this level of regeneration, push the level of impairment down a little bit. And why is that important? I told you about individuals living with tetraplegia who find that hand and arm function really is critical for their quality of life. And so this is a table showing the capacity for activities that are related to daily living for individuals with spinal cord injury, things about bowel and bladder function. But bed mobility and the ability to transfer oneself. Pressure relief. So pressure sores are a very serious concern for individuals with spinal cord injury that have limited movement, and cause infection and sepsis. Feeding themselves. We saw a video of somebody practicing feeding themselves with a stimulator. Dressing and grooming themselves, bathing. These are things that somebody with like a cervical 5 injury is going to have some severe problems with. But if you could just move that function down by one level, suddenly they're much more independent. Their quality of life is dramatically improved. So that's really, what we're aiming at with this here. And here's some examples of individuals that have had nerve transfer. Now, Dr. Brown is asking this individual to lift her left arm, which is impaired. This is not the one she had the transfer surgery on. She had the transfer surgery on her right arm. Now I asked her to lift from the elbow, and you can see now she can move it. Okay, the other arm's impaired. We're going to look at the hand of an individual with transfer surgery. This is the impaired hand first, asking him to make a grip, and he uses this tenodesis grasp where kind of pulls his wrist back and his wrist closed a little bit. But now you can see the transfer side when you ask him to make a grip, he's able to close his fingers. He's able to close his digits here. And so this hand would have looked like the other one prior to the nerve transfer surgery. And now you can see he's asking him to grasp. He can use his thumb a bit and grasp. It's not perfect, but it's a huge step in the right direction. So Sebastian has generated a mouse model of nerve transfer in conjunction with Dr. Brown. It's very rudimentary. We take the musculocutaneous and graft it to the median nerve in order to shift function from the proximal forelimb, from elbow flexion to the hand. And so this is an ongoing project. I'm not really going to get into great detail about it. I know I need to cut things short, so I'm not sure I'm going to get to much in terms of our current research, but I do want to mention, what we're interested in and why. So one of the critical aspects that's been overlooked, and this is sort of what drove the award that Christine mentioned at the beginning, this new innovator award from the NIH, is we get very caught up on the spinal cord in terms of spinal cord injury research. How to make new axons, how to make things grow, how to make things regenerate, and for good reason, right? But one of the things that hasn't really been focused on is what the role of the brain is in all this, and what the role of the cortex is, because no matter what changes you make in the spinal cord, if you don't have some kind of commensurate changes in the brain, the body's not going to be able to figure out how to, you're not going to be able to figure out how to use that rewired spinal cord, whether it's, this local plasticity or whether it's trying to engage these bypass circuits. So let's see. Christine, hopefully it's okay if I use a couple more minutes to go through some very basic slides, hopefully. In order to do that, we use a virus injection into the spinal cord, and we want to look specifically in these neurons that project to the spinal cord. So if I've lost you at any point in the next few slides, don't worry about it. I already covered most of what I wanted to talk about today. This is just sort of the new and flashy stuff that we do in my lab. So again, completely in my own self-interest to tell you about this. But what we can do with this virus is we can express different genes, and this one is a calcium indicator. So I told you that within neurons, the changes in ion levels and the electrical activity is what drives communication within the nervous system. So calcium changes really dictate the electrical potential of a cell. And what we're looking at here are individual cells, neurons, corticospinal neurons project from the cortex of spinal cord. We're looking at them flash as calcium is released within these cells. So that's a measure of their activity. And what we can do then, and this is amazing work by Najet Siraj and Francesca Marino, is we can pick a few of these cells out and we can record their activity. Those little flashes, we can record these traces here as they go up and down while the animal's doing a behavior. This behavior is marked by these vertical lines here. So either they're successful in this behavior, which is a red line, and they get a reward, they get water to drink, or they're unsuccessful in these vertical gray lines. And you can see this is just an example of 5 independent cells. And this is sort of what we look at in terms of the behavior. Oh, I got the wrong one. Here we go. We use this isometric pull task. The animals have to reach out and grasp a lever and pull on it. And what we do is we teach them to pull and release this lever. We teach them a skilled version of this task. And we also have an unskilled version of this task, very simple task. Basically, they just have to pull on this knob above a certain, with a certain strength, and then they can get some water in reward. And so again, I'm going very rapidly to wrap things up, but the animals learn this over time. They learn this skilled task over the period of 5 weeks here. We have early, middle, and late stages. In the unskilled task, they learn it pretty quick in about 2 weeks. So this, they know how to do it right away. And if we then look at how consistent the behavior becomes as they become experts at it. Again, if you are glazed over and I'm going too fast for you, don't worry about it. We'll converge at the end. Basically, on the bottom here, it says pull-to-pull correlation. That just means the animals become more consistent, right? As somebody learns how to shoot free throws, they get better at it. The movement becomes more consistent. They start making more shots, being more successful. So as the animal pulls more and more and refines this movement, each pull becomes more consistent with the last one. That's this pull-to-pull correlation. So we have something similar on the vertical axis here, activity correlation. That's just if the cells are becoming more and more similar over time in terms of how they're flashing, those little bright flashes of light. So they're activity patterns. And we can see that they increase over time. So as the animals become more consistent in their behavior, then their activity becomes more consistent. Whereas in the unskilled task, it's kind of flat. It doesn't really have that same increase. If we injure this, so if we take out this entire corticospinal circuit, the animals can't do the behavior very well anymore on the unskilled, but they can do this very simple task. So this is that circuit I was telling you about, that there's differences between rodents and primates. This is something that's really important for us, this circuit, in order to do our day-to-day functions. And we have to develop these fancy tasks to address it in rodents so that we can understand it. So really quickly, injury disrupts this. I'm not going to touch on that, but these are the people that make it all possible, the people that have done this work. Sebastian Hara is on the right here. Najat Siraj did a lot of the imaging along with Francesca Marino. Najat Siraj in the center and Francesca just to her right, or just to her left, our right. There's a number of others I don't really get to tell you about. Our collaborators, Justin Brown, who we're working with in terms of the surgery, and Kathleen Friel, who's then looking in these individuals and doing rehabilitation with them. And we're looking at how their brains incorporate these changes. Amanda Bernstein is really critical in those studies. Number of other people that I don't really get to touch on, but our funding, I need to point out, while we have gotten funding from the NIH and from the Nielsen Foundation, without the New York State Department of Health, SCRB, my lab wouldn't exist. And so I really do have to thank them for giving me this opportunity, along with Raj and all the people at Burke. Sergeant Paul Richter was one of the people instrumental in getting this started, and unfortunately passed this year. And so with that, I'll take questions.
Edmund, thank you so much for sharing your groundbreaking work with us today. It was a wonderful presentation. And yes, the questions have already started rolling in. So maybe you can take a sip of water and get ready to talk.
Sorry, I went longer than I intended. I just got really excited to try and show Najat's work at the end there.
We share your excitement. The first question is from Jill Cooper. Do you have international cooperation or collaboration with other researchers on SCI treatments to see if pain is primary consideration on daily functioning? Are there any non-invasive treatments up and coming?
Are there non-invasive treatments specifically for pain? I'm not I'm not aware of non-invasive treatments for pain. So pain was covered by Dr. Willis. And I think most of the pain research is focused on therapeutics and drug delivery. But one could imagine that things such as this stimulation, this electrical stimulation, may actually affect pain. One of the things that Sebastian is working on is trying to understand how different patterns of stimulation will affect different sensory neurons, the ones that mediate movement, the ones that are important for now I say movement. Movement is motor and sensory information combined. So if you reach out and if I reach out and pick up my glass of water, you know, I don't have to look at it in order to drink from the water. I know where it is because my body is telling me where it is. My body's telling me how heavy it is, how full it is. You know, there's a lot of information that I can get without looking at it. And so those neurons are ones that have been the focus of these regeneration studies I told you about. They mediate movement. There's smaller neurons that mediate pain. And so different patterns of electrical stimulation will have different effects on them. So potentially, you know, it's possible that some of these non-invasive stimulation patterns could affect pain. I don't know of work that's specifically looking at it. We don't have any international collaborators at the moment. Most of our collaborators are stateside and focused on movement again. So thank you.
Thank you. Also, too, just a quick reminder, we're going to be wrapping up pretty soon, but we do have some time for questions. So please type your questions in the chat window and I'll read them to Dr. Hollis. Dr. Hollis, does spinal cord injury affect lifespan?
It does, yes, and that's a very real consequence. So I don't have the numbers in front of me, but if you go to the National Spinal Cord Injury Statistical Center, I had a link to one of their slides in here earlier. Lifespan is attenuated for people with spinal cord injury. It really depends on the age and the severity of injury, but I mean, complications that you get from things such as bed sores and infection. I mean, these are very real issues, and these are issues that people are, you know, people are angry about. They want treatments to drive this. So yeah, it's mostly from complications. I mean, pneumonia. You could think anybody who's had to be a caregiver for somebody with spinal cord injury or stroke or any kind of paresis knows, you know, there's difficulties and problems getting around. So pneumonia is a very real concern, and especially in the age of COVID right now. You know, there's a lot of complications that come with not having a functional nervous system.
Thank you. And is depression a common occurrence with spinal cord injury patients?
I don't know the numbers. I don't know. I imagine so based on personal experience with people with paresis, but I don't know the numbers, and I'm sorry, I can't answer that question. But certainly engaging people and giving people hope is important. And so, like I said, there's a balance in terms of hope and motivation versus overpromising. And I think that's a very fine line that we walk as scientists.
Absolutely. In the video that you showed with the arm stimulation and eating, is that technology applicable perhaps to somebody with Parkinson's disease?
So Parkinson's disease, people have looked at deep brain stimulation for that. I am not as familiar. I don't know that the transcutaneous stimulation, not just on the surface, would be sufficient. Parkinson's disease is a degeneration of neurons in the brain itself that are not these large output neurons, but more neurons that shape the movements. And so I think there's been progress with deep brain stimulation, but I don't know the full extent of it. So I don't know that this transcutaneous would be enough possible. You have to ask Dr. Edgerton and Dr. Gadd work on this.
Interesting. Thank you. And last question. Somebody was very curious about this, and so am I. What is the difference between a skilled and an unskilled task? You showed your mouse models.
Yeah.
And how can you relate that to human behavior?
So that's right. That's kind of the thing. So, right. So an unskilled task is really something that's very simple, and it's important because in a rodent, if you like these animals where we've completely cut this major descending pathway that's mostly motor, but some sensory as well, these corticospinal axons. If I gave you a mouse that had no corticospinal tract because of this injury, you probably wouldn't be able to tell the difference between that mouse and a normal mouse. If I put one cage, one in the other, and I said, which one's injured? Yeah, you're probably not gonna be able to tell because the animals can ambulate, they can walk about, they can do mouse things, they can eat their food. But in a human with a corticospinal injury, it's gonna be very different. There's gonna be a lot of weakness. You know, people don't get just corticospinal injuries in humans, as I showed in the non-human primate schematic. The corticospinal tract is nestled in what's the dorsolateral spinal cord. It's got other axons on top of it. It's you know, it's not just going to be a clean cut of that. There were some studies looking at what was the underlying, I don't remember what the underlying condition was, but they ended up doing these pyramidotomy injuries in humans with to transect part of the spinal cord. I'm sorry, I don't remember what the underlying condition was, but essentially these people would regain the ability to walk after having a unilateral injury to the corticospinal tract. And it took some time, but it was thought to be mediated by the other corticospinal tract. And when there was another lesion of the other corticospinal tract, then those individuals could no longer walk. It was a therapeutic intervention, and I don't recall what it was for off the top of my head. So, you know, how much of that was related to the sort of the plasticity mechanisms, like I showed of Dr. Carmel's work with Jack Martin. You know, how much of it is the ability to rewire, and how much of it is just the ability of other. We have lots of other motor pathways that come from the brainstem, that come from the midbrain. So we have other descending pathways that can drive behavior, and this makes sense. You know, the corticospinal pathway is really, it's engaging our highest level of processing. So this is sort of the pathway that goes on when you're composing an email or something, and you have to figure out, oh, you know, which keys am I looking at, and where am I getting to? But, you know, say when you're driving a car or something, you know, and you're just engaged in the moment. You're not necessarily considering about how much turn it becomes second nature. And so you have motor programs and motor abilities that don't necessarily require your full attention to detail. You know, maybe when you first get behind the wheel, when you're 16 and you're learning, you overcompensate. You know, your lane changes are not smooth, things of that nature. But then once you get it down, it's not something that you think about. And so there are other pathways that drive these behaviors that can take things from being sort of skilled and engaging the cortex to sort of unskilled. So there's even a movement between what might be considered skilled and unskilled. We're addressing it in the mice because what has been shown in the past is that with unskilled behaviors, so simple lever press behaviors, is that the brain is very plastic and can change and learn and rewire. So when somebody learns how to play the piano or learns how to type on a keyboard, representations of their digits will increase, that their fingers will increase as they learn how to play the piano. So that the part of their brain that is engaged in that will be larger. And in a mouse or rat that learns how to do a skilled behavior, they'll see that same sort of enlargement of the areas related to movement. So as the animal reaches out and picks up a small piece of food or something, if you train an animal to do that over and over again, then you have a larger representation of the wrists and digits. If you train them to just press a lever, it doesn't do anything to their brain. And so we're trying to figure out how engaging the corticospinal tract, which is critical for the skilled movement, and the cortical changes that we see and that we think we need to see after injury, how they're related. So that's why we focus on these skilled and unskilled tasks.
Thank you for that explanation.
Well, that's very long.
No, it was fantastic. Thank you. And again, Edmund, thank you so much. I know how busy you are. Thank you for sharing your time and your expertise and your study with us. For all of those who are interested, please keep an eye out for Dr. Hollis's study. You can also see it going onto the Burke Neurological website. And also our patient registry is there as well if you know anyone who could benefit from clinical trials at the Burke Neurological Institute. We at the Burke Neurological Institute would like to thank everybody for their time and participation today. We will email the recorded webinar to all the participants who registered. Please share with anyone who may be interested and follow the BNI social media monthly communications for more updates and webinars. Lastly, we'll be sending out a webinar survey. Please give us your feedback so you can help us help you. Have a great week and stay well, stay safe, stay informed. Thank you and goodbye, everyone.
Thank you, everyone.